I'm Nairin S. Mantawid. I'm the leader of this group. So for today, I will discuss about meiosis. Meiosis is a special type of a cell division of germ cell in sexually reproducing organism used to produce the gametes such as a sperm or egg cell. This is a process where a single amount of cell can divide twice to produce 4 cells containing half the original amount of genetics. Diploid is a 2 set of chromosome in human 2 and in equal to 46 all body cells other than gametes is called diploid. While the haploid is the 1 set of chromosome in human number, it is equal to 23 gametes or sperm cell and ova are called haploid. Importance of meiosis in human. Meiosis is very essential to produce all the organism via sexual reproduction. It is responsible for the fermentation of egg and sperm cell of humans. Stage of meiosis. Meiosis is the split into 2 phases, meiosis 1 and the meiosis 2. In each of the phases, there is a prophase, metaphase, anaphase, and the telophase. In meiosis 1, these are known as prophase 1, metaphase 1, anaphase 1, and the telophase 1. While in meiosis 2, they are known as prophase 2, metaphase 2, anaphase 2, and the telophase 2. Meiosis is known as reductive division as the cell of are reduced from being diploid cell to being haploid cells. And the next is Aludin Suhaiber discuss the prophase 1 and the interphase 1.
Prophase 1 is the longest phase of meiosis with 3 main events occurring. The first is the condensation of chromatin into chromosomes that can be seen through the microscope. The second is the synapsis or physical contact between homologous chromosomes and the crossover of genetic materials between these synapse chromosomes. Interphase is the longest stage in the eukaryotic cell cycle. During interphase, the cell acquires nutrients, creates and uses proteins and other molecules, and starts the process of cell division by replicating the DNA. Interphase is divided into 3 distinct stage 1, gap 1, and synthesis, and gap 2, which are discussed below. The purpose of interphase in all cell types is the prepare of cell division, which happens in a different stage of the cell cycle, depending on which species of organism is dividing. The function of the cell during interphase can vary widely. Some cells, like neurons, do not replicate their DNA during interphase, but interesting phase or sequences instead. The stage can be seen in the graphic below as the GO stage. In the stage, the cell will exist without dividing until the cell dies. Other cells like skin is divided a lot. Crossing over is a biological occurrence that happens during meiosis when the paired homologs or chromosomes of the same type are lined up. And it is this crossover that lets recombination across generations of genetic materials happen, and it also allows us to use that information to find the location of our genes.
The next is Ibrahim Baragir discuss the end of the phases 1 and the metaphase 1, anaphase 1, and the telophase 1.
Let's proceed to my topic. So, the first is metaphase. Metaphase 1. So, metaphase is encompasses the alignment of paired chromosomes along the center. Metaphase is ensuring that 2 complete of copies chromosomes and are present in the resulting of 2 daughter of cell of meiosis 1. So, metaphase follows prophase and preceded by anaphase 1. So that's all for metaphase 1, and let's proceed to the next. The next is anaphase 1. So this stage is characterized by movement of chromosomes to both poles of meiotic cell via. So anaphase is the third stage of meiosis 1 and followed by prophase and metaphase 1. So this stage is network known as the spindle apparatus. Then this mechanism is separates homologous chromosomes into two separate groups. So that's all for anaphase. So let's proceed to the telophase 1. So telophase 1 is phase when the chromosomes finish moving to opposite end of the cell. Then be followed by cytokinesis. Cytokinesis is producing two daughter cells. After the cytokinesis, the two daughter cells would have genetical different chromosomes after the meiosis 1. So that's all for telophase. Let's go to the last one. So the last one is mitosis. Mitosis is begin at prophase with thickening and coiling of the chromosome. The end of prophase is marked by the beginning of the organization. End of prophase, a group fibers to form a spindle and the disintegration, the nuclear membrane. So that's my last topic, and that's all. So thank you very much for listening. Then have a good day. Bye. Peace.
And the next is Ibrahim Almira will discuss the prophase 2 and metaphase 2 and anaphase 2.
Prophase 2. During prophase 2, the nuclear envelopes dissolve and new spindle fibers forms. Prophase is the first phase of mitosis and also the longest. In this phase, the cell chromosomes begin to condense into tightly coiled, compact structures. This enables the chromosomes to move easily through the cell without getting tangled. So let's proceed the topic number 2. The number 2 is metaphase. Individual duplicated chromosomes align on the equator. One chromosomes for spindle fiber attached by means of kinetochore of centromere. Centriole has reached the poles. Metaphase is the third phase of mitosis, that process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. There is an important checkpoint in the middle of mitosis called the metaphase checkpoint, during which the cell ensures that it is ready to divide. Let's proceed the topic number 3. So the topic number 3 is anaphase 2. Sister chromatids are separate from each other. Anaphase is a very important stage of cell division. It ensures that duplicated chromosomes, or sister chromatids, separate into two equal sets. This separation of chromosomes is called disjunction. Each set of chromosomes will become part of a new cells. Anaphase, the sister chromatids separate from one another and are pulled towards opposite poles of the cell. The microtubules that are not attached to chromosomes push the two poles of the spindle apart, while the kinetochore microtubules pull the chromosomes towards the poles.
The last one is Ismail Asnaira will discuss the telophase 2 and the error of meiosis.
My name is Asnaira Ismail, and my topic for today is telophase 2. Telophase 2 is the stage in meiosis 2 that follow after anaphase 2. This will then be followed by cytokinesis, wherein each of the two cells produced from meiosis 1 will give rise to the two daughter cells, resulting in a total of 4 genetically dissimilar haploid cells. The chromosomes decondense and lengthen. Final results. Cytokinesis splits the chromosome sets into new cells, forming the final products of meiosis, 4 haploid cells in which each chromosome has just one chromatid. In humans, the product of meiosis are sperm and egg cells. Errors in meiosis. Non-disjunction occurs when homologous chromosomes, meiosis 1, or sister chromatids, meiosis 2, fail to separate during meiosis. An individual with an error in chromosomes number is described as aneuploid, a term that includes monosomy, loss of one chromosome, or trisomy, gain of an extraneous chromosome. When non-disjunction occurs in meiosis 2, cells divide normally during anaphase 1, homologous separate correctly, but sister chromatids fails to separate during anaphase 2. As seen in the diagram above the left, one cell shows trisomy 2n plus 1 and the other shows monosomy 2n minus 1.